And so before that happens, we're going to have two speakers. The first of which will be Tom. Is it Nye?
Yeah.
It's Nye. So yeah, Tom Nye, and he's going to tell us about seeing the wood for the trees. Thank you.
So you use the expression "you can't see the wood for the trees" to mean that you're getting lost in the detail of a situation. You can't see the sort of salient facts because you're getting overwhelmed with information. You need to step back and see what's going on. And I like this expression because it applies to a situation in phylogeny. So a lot of the time when you perform a phylogenetic analysis, you get lots of different trees out. So what I'm going to be talking about is a situation where instead of having a single tree, we have lots of trees on the same set of species. And this can be for various different reasons. So it can be because we're interested in variability in the data. So we might have a collection of trees that comes, for example, from bootstrapping, if we're doing maximum likelihood estimation. Or we might have a collection of trees that represent some sample from a posterior distribution. As Tandy said earlier, there's also a situation where we might have different trees for different genes. So we might have a whole bunch of different trees, one coming from each gene. And the question is, how can we summarize and represent all this information? And what I'm going to be talking about in particular is how can we compare different tree topologies. It's actually quite a problem if, say, we've got 100 trees on 1,000 species, just to work out which trees have the same features, which trees differ, where they're different. So it's essentially a sort of visualization problem that I'm going to be talking about. So there are existing methods for doing this sort of thing that you're probably familiar with, like consensus trees or using a network instead of a tree to represent sort of conflicting evolutionary histories. There's also another couple of methods which I think are worth mentioning because they're relevant to what I'm going to be talking about. The first is multidimensional scaling. So this was published in Systematic Biology a couple of years ago. This is a method where you essentially project tree space into the plane. And so you represent your trees as points in tree space. And then there's a second method which is similar to what I'm going to talk about in its approach. And that's a sort of clustering approach which was published in Bioinformatics a couple of years ago. So that's where you cluster trees according to their topology and you take consensus of these clusters that you've obtained. So that's rather similar in method to what I'm going to be talking about here. Okay, so here's a toy example. 4 trees. How are these related? Well, you can see that we've got species A and B next to each other here, and then A and C next to each other here, but otherwise they're completely different. So one way to represent these trees is by a tree of trees. Okay? So here we've got our 4 species trees, 1, 2, 3, 4, and then in the middle I've drawn a sort of meta tree, or a tree of trees, which joins them together according to how similar they are. Okay. So what I'm going to be talking about today is how we can construct meta trees. So there's an obvious objection to this, and that's that tree space does not intrinsically have a tree-like structure. Okay? So in some ways, doing this might not be a very sensible thing to do. On the other hand, there are certain advantages to doing this. So a meta-tree will tend to cluster similar trees together, for example, and conflicting histories are going to show up as different groups on the meta-tree. Okay, so we might have different clades on the meta-tree that might represent, for example, different modes in a distribution, or maybe outliers. So if there's a tree that's very different from all the other trees, it will show up as an outlier on the meta-tree, enable us to detect these unusual sort of topologies. And moreover, having a tree, of course, is a convenient form of visualization. We all know how to interpret trees, how to look at them, and so maybe a meta-tree is just an easy way of getting a handle on this complicated data set. So here's a definition of a meta-tree. I'm going to be working with a fixed set of trees, and I'm really only interested in their topologies. And they're all on the same set of leaves L. Okay? And a metatree is an unrooted tree that has n vertices, so n leaf vertices. So there's one vertex for each of the trees. And at each internal node of my metatree, there's going to be a species tree. Okay? So the leaves on my metatree are associated with the original collection of trees. I'm going to give a score to every metatree, and I'm going to be looking for an optimal metatree. So I'm going to score metatrees by adding up edge scores. So I'm essentially scoring each edge, and then I sum the edge scores. And the edges are scored according to the similarity between the tree topologies at each of the edge vertices. Right? So I have this sort of metric D that says how similar my two trees are, and then I give the edge that score. So there are a number of different metrics that we could use there. So for simplicity and for algorithmic reasons, I'm going to concentrate on the Robinson-Folds metric. So here's a very quick sort of recap, which you probably already know about, to do with the Robinson-Folds metric. So a split is a bipartition of a set of leaves on a tree. This is a tree rather than a metatree. Trees consist of compatible sets of splits. There's something called the majority consensus of a set of trees, which is a tree which contains the splits in strictly more than half the trees. And then the Robinson-Folds metric basically counts up how many splits you've got in one tree but not the other. So it consists of the number of splits in tree A but not tree B, plus the number of splits in tree B but not tree A. So there's a sort of analogy here between constructing a metatree and parsimony for DNA. So suppose we use this Robinson-Folds metric. We can then represent tree topologies by strings of ones and zeros to mean absence or presence of splits. So could we apply our parsimony algorithms to build an optimal metatree? And the answer is no, because we need each of these strings to be a compatible set of splits. And in general, if we apply this sort of algorithm, we don't get a tree-like set of splits. And the general computational problem here is that we've replaced this set of characters of the DNA letters with the set of tree topologies with leaf set L, which is a huge space. So computationally, this is an awful problem. It looks like metatree construction may take longer than actually constructing your trees in the first place, which is ridiculous, because this is supposed to help us. So what can we do? Well, there's a very nice property of the Robinson-Folds metric, which I'll explain here. So if we consider a metatree with a star topology, so here's a metatree, the central node I'm going to denote T0, and then we've got our collection of trees around the outside. Well, we can write down the score for this configuration. It's the number of edges with a split P at one end, but not at the other, summed over the set of splits. And this score is minimized by taking the majority consensus at the middle. And this result is called, it's usually referred to as saying that the majority consensus is a median tree. Okay? And this is a particularly nice property because we can optimize here. This optimization occurs split by split. So we consider each split independently. We don't have to consider their compatibility. We do the optimization, and then at the end of this process, it just turns out that the collection of splits in the middle really is a compatible set, and we have a tree. So can we generalize this nice result to something that will help us construct metatrees? Well, this property extends to every vertex on the interior of a metatree. So here I've drawn a maximally resolved vertex inside a metatree. And if we just think about whether to include a split in this topology or not, if we have two neighbors that contain the split P, but one neighbor that doesn't, then you can see the score is minimized by including the split P at this interior vertex. Similarly over here, if we've got two neighbors without the split, one neighbor with the split, then the score is minimized by not including P in the interior vertex. So what you have here is that this majority consensus property that we saw for the star meta-tree phylogeny extends to give us a local optimality criterion for any interior vertex on an optimal meta-tree, so a meta-tree with minimal score. So this inspires the following heuristic algorithm for meta-tree construction. So I call this the Meta-NJ algorithm because it's quite similar. It's sort of inspired by neighbor joining. So what I have here are, I'm drawing meta-trees. It's always useful to say whether I'm drawing meta-trees or trees at any stage because it's quite easy to lose that. So this is a meta-tree here. And I carry out the following algorithm. I choose two vertices A and B. So those are two tree topologies. And I decide to agglomerate them. So I draw this configuration where I've split off trees A and B. I've got these new nodes X and Z. So I then choose X and Z to be the majority consensus of their respective neighbors. So X is going to be the majority consensus of A, B, and Z is going to be the majority consensus of its neighbors. I then score this configuration, and I go through all different pairs A and B, and I choose the pair A and B that minimizes the score of this configuration. So once I've chosen that, I kind of throw away A and B, and I work with this tree, and I continue to sort of work with that tree. So the algorithm repeats from that stage. So at each stage, I'm essentially sticking two nodes together, working out the majority consensus to obtain the topologies X and Z. Okay, so there are a few interesting features for this algorithm. The simultaneous equations for X and Z could be solved simultaneously. Okay, it's not obvious. And you consider each split independently to solve those equations. So it's computationally really quite fast. Each vertex in the resulting meta-tree satisfies this local optimality criterion. And there's a few other points. You can obtain multiplications because you sometimes get zero-length branches. There can also be ties in the score when you just pick one of these agglomerations at random. So I'll show you quickly some examples of the sort of thing you can do with meta-trees. This is a fairly well-known data set published in Nature, so it consists of some trees for 8 species of yeast. There are 23 different topologies obtained, and I've just drawn the consensus network there. And it's interesting just to point out that these two species, there's some sort of lack of resolution there and lack of resolution down here. When we draw the meta-tree, though, you get a completely different picture. So your focus goes from splits to trees. So here's the meta-tree. You can see the 23 different topologies. The branch lengths are drawn so that they're proportional to the scores. So meta-trees are much easier to sort of interpret when you know or when you can see what topology is associated to each vertex. So for example, here, associated to this sort of outlying vertex, we have this topology. So the software I've written allows you to click on a vertex in the meta-tree, and you can see the associated tree topology. So this topology 11 is a sort of outlier. There's something weird going on with this particular gene, okay, because its topology is radically different from all the others. None of these splits, I think, are found anywhere else in the meta-tree. Similarly, you can kind of interrogate other internal vertices to try and work out why your meta-tree has this particular appearance. So here, this node here is associated with this topology, and these two species were two that you saw on the consensus network, which were relatively unresolved. And this split of these two species nicely splits the meta-tree into two parts. So the split is present down here, but absent from these trees up here. Similarly, you can interrogate other vertices. You can see that this split is contained in all of these nodes of the meta-tree, but not over here. So by interrogating your meta-tree, you get an idea of why it's being constructed in this particular way. The second data set I've looked at consists of 10 orthologous genes in 14 species of fish. So there's the metatree for these different genes. It's rather long and kind of straggly. You've got these long exterior branch lengths that shows that there's a high degree of conflict between the different tree topologies you're getting out for each gene. So at the very worst situation, you'd actually have a star phylogeny here, which would show sort of a maximum degree of conflict between your different gene topologies. So it's interesting to ask the question, to what extent is this incongruence caused by a lack of phylogenetic signal in each gene sequence, or whether there's genuine evolutionary differences between these genes? So what I've done is to just do 10 bootstrap replicates. You could do many more, but it's easier just for the point of view of visualization to do a relatively small number, and then see whether the replicates for each gene form clusters on the metatree. So there's the metatree. It's quite hard to see what's going on there. There are 100 leaves there, 10 genes, 10 bootstrap replicates for each gene. So on this slide, I've just colored in each gene. So there's just 4 genes that I've colored in. It's not come out very clearly. So the replicates for each gene have a different color. So this is one gene, red, blue, you've got another gene. And you can see that these genes are sort of, these replicates are spread out around the metatree. So it seems to be indicating, rather than a genuine conflict between these genes, more a sort of lack of information in each of the individual gene histories. Okay, so just to summarize, I think, I've attempted to represent collections of trees using trees of trees or metatrees. Finding an optimal metatree is computationally very difficult. So I've presented a heuristic algorithm for constructing these. And in particular, it constructs metatrees so that each vertex is the majority consensus of its neighbors. Examples show the sorts of things you can use these metatrees for to find out about your data set. So just to finish off, thanks very much to Wally Gilks for his input into this work, and also to Antonis Rockhouse and Chenghong Li for providing me with the data sets I've talked about. This software is available online, and if you're at all interested in what I've spoken about, I recommend having a look at that because it really helps you understand the advantages of using this sort of method. Okay, thanks very much.
Questions, please?
So would there be a limit on how many trees you could usefully use? This software could be usefully used.
No, there isn't really. It's because it's heuristic. Because I made simplifying assumption, it's quite quick. I mean, it's n cubed in the number of species. And so it scales quite well. Yeah.
I mean, to construct these gene trees, you needed alignment for the genes.
Mm-hmm.
Which alignment methods did you use, and did you play with different alignment methods?
No, I've received the data from the authors of these papers. So I've basically received trees from them, and I've analysed those trees. So I haven't built the trees myself.
Because, I mean, you could begin doubting all the gene trees as they are so conflicting with each other in the last example.
Right. Okay.
But you never checked whether these
I'm not
are
involved. I've inherited the data from other people. So, yeah.
One more.
Okay. Have you tried really testing if just by maximum parsimony or something you get incompatible substructures, or if just by chance that will never happen for real data?
Sorry, could you explain that again?
On the inner nodes, you could simply do parsimony, but you say that this wouldn't then correspond to trees. Okay?
Right.
And you could just run it anyways and see if what comes out of it is a tree.
You could do, yeah. I've not tried that, but in general, your set of splits corresponding to internal nodes, there won't be a guarantee that they'll actually
No, not a guarantee, but
be tree-like. Yeah.
I'd happen anyways.
Yeah.
Just wondering if you could sort of imagine that you could have many, on a small data set, you could have many different possible trees, and that maybe the internal vertices are actually a better estimate of the real tree, because they're somehow summarizing what all the other trees are saying, but or what is in common with all the other trees. I was just wondering if you thought along those lines.
Yeah, I mean, you need to be careful because what I'm presenting is not at all statistical. There's no statistical assumptions. It's heuristic. It's just a way of presenting your data. So it may be producing interesting consensus trees, but what they mean, I'd be very careful about.
Okay, thank you very much.